Phoebe Cohen is our speaker this evening, and she is an assistant professor in geosciences at Williams College. She applies a wide variety of microscopic and microchemical techniques, combined with complementary data from field-based stratigraphy and sedimentology to reconstruct ancient organisms and ecosystems.
I grew up in Newton. And I used to have my birthday parties here at the Arboretum. When I was a kid, we'd have picnics and play capture the flag. I used to come here in high school. I spent a lot of time here as a youngster. So it's really wonderful to be here and to be talking to you tonight. So what my talk is going to be about today is mass extinctions. So this is sort of a new area for me. I'm going to be giving you a broad overview of what mass extinctions are and talking briefly about the work that I'm doing on one of these mass extinctions. Okay, so first a little bit of background before we get going into the nitty-gritty. So I'm a paleontologist. When most people hear the word paleontology, they think dinosaurs. So they think dinosaurs like this, or they think dinosaurs in the ground and someone with a little paintbrush, brushing away these beautifully articulated bones. Or maybe you think of things like trilobites, which are these extinct arthropods beautifully preserved here. So in reality, paleontology is the study of all extinct life, from single-celled bacteria to dinosaurs. And usually, we view that life through the fossil record. So through the physical remains of organisms, their shells, their bones, other remains that they leave behind. And the goal of this work is to reconstruct the history of life on Earth. So, just a small task that we set out for ourselves. So today what I'm going to be doing is talking a little bit about geologic time and how we measure the diversity of these fossils that I just described to you through time. And then I'll talk about what mass extinctions are, the three largest mass extinctions. Then I'll talk about something we call a mass depletion, which is the event that I'm working on. I'll talk very briefly about whether or not we are currently in a mass extinction. And then I'll try to end on a more hopeful note, talking about the changes that mass extinctions have had on life through time. Okay, so a very brief overview of Earth history. So the Earth is about 4.5 billion years old. GA means billions of years ago. And you can chunk up this Earth history into three pieces. The Phanerozoic, which means visible life, which is where we are now. The Proterozoic, which is the time period of not so visible life, which is what most of my research to date has been on. And then the Archean and the Hadean, which is either there's no life or it's extremely confusing and I don't want to deal with it. And so here are some representative fossils from these three time periods. So things like trilobites, which you can see with your eye. You can walk up to a rock and see a trilobite. Not so visible life would be things that are microscopic. And then confusing and not so visible life, things that may be fossils, but we're not quite sure. So the Phanerozoic over here is where most of our fossil diversity comes from. All right, this is the time period where we have animals. So we have trilobites and we have dinosaurs. We also have flowering plants and land plants in general. So this is a ginkgo. If you turn around, you'll see some beautiful ginkgo leaves hanging from that string there. Ginkgos are super cool because the leaf morphology has changed basically not at all in 145 million years. Okay, so the fossil that you're looking at here is basically exactly what you're seeing back there. Perfect little teachable moment right there. And lots of other things, insects and starfish and what have you. So what we'll be talking about today will be solely in the Phanerozoic. Trying to pinpoint a mass extinction in this time period would be really interesting, but our fossil record isn't good enough for us to be able to do that yet. When we talk about extinctions, we're talking about statistical arguments using large numbers, and so we need lots and lots of fossils to make a strong argument. And so all the mass extinctions I'll be talking about are over here in the Phanerozoic. Okay, so how do we actually document a mass extinction? So let's say each of these black lines represents a fossil taxon, okay, so a species or a genus. The beginning of the black line over here represents when the group shows up in the fossil record, and then the end of the line indicates where it goes extinct. Okay. So if we take these lines and we chunk them up into time bins, and then we count how many lineages are in each bin, okay, so there's 3 here, and there's 4 here, and there's 5 here. We get these numbers down here, and then we can plot those. And that's a diversity curve. It turns out that there's a lot of ways of counting diversity, which I'm not going to talk too much about, but this is the most simplistic form. All right, we're just seeing how many fossils are in each of our time bins and counting them up, and then seeing how they change through time. Okay? So the first person to do this actually did it over 150 years ago. And this is the curve that Phillips came up with. He said, OK, he basically was only looking at fossils that were found in the United Kingdom and parts of Europe. And he made this curve, right? So diversity goes up and down a little bit, up, down, up, down, up, down, up, down. If we then move to the figure that we use today, it has some remarkable similarity to Phillips' figure. So now this is on its side. Okay, so we just sort of flipped it around. This is 541 million years ago, and this is today. And this is called the Sepkoski curve, after Jack Sepkoski, who's a very famous paleontologist. And Jack published 2 papers. The first one was called 10 Years in the Library, and the other one was called Another 10 Years in the Library. And he did this in the 70s and 80s, and he was at Chicago at the time. He went to the library and he documented all of the diversity of marine, mostly invertebrate fossils. So clams and snails and starfish and corals and things like that, which are the most sort of abundant and diverse and robust fossils that we have. And so this is the curve that Jack came up with. The colors here are just these sort of informal groupings that we use to talk about different groups of organisms, so don't worry too much about them. So this is what we call the Sepkoski curve after Jack's hard labor. And apparent in this curve are 5 inflection points. All right, and so these are what are called the big 5 mass extinctions. And you can see them pretty easily on this graph. You can see this one, which is certainly the largest, and we are over here somewhere. OK, so I just showed you these inflections, but what makes them mass extinctions? And one of the questions is, things are always dying, things are always going extinct. What makes a mass extinction mass? So it turns out that if you ask different people, you'll get different answers. But the general rule is that it's a loss of over 75% of species in a relatively short period of time. So what that period of time, relatively short geologically speaking, so like 1 million or so years. Okay? It's also where rates of extinction are much higher than the time intervals before or after. Okay? So you might be saying to yourself, well, how do you really distinguish that? So let's look at it a little closer. So here's what diversity looks like. This is measured in number of families. Okay, so like who remembers the King Philip thing? All right, so if you do the Linnaean taxonomy, family is sort of in the middle. So number of families, so this is like just raw number of things that are alive at any point in time. We can also look at it in terms of the rate of extinction. Okay, so in each time bin, what's the rate at which things are going extinct? And if we do this, we get another figure, also made by Jack Sepkoski. And the little pink circles here are highlighting our big 5 mass extinctions. So during these extinction events, extinction rates are elevated above what we consider to be background extinction, which is all this stuff here. So again, this is 600 million years here, and this is today. So we're moving this way. Okay? So they stand out in terms of rate. Now, it turns out there's some other fun, interesting things that sets mass extinctions apart from background as well. And this is more recent work done by some friends of mine, John Payne and Seth Finnegan. And here we have, again, the beginning of the Phanerozoic here and today. And this is sort of a tricky chart. But what it's showing is that if you're above the 0 line here, it means that if you're a group of organisms that has a broad geographic range, then you are protected from extinction. Okay? If you're at the 0 line, it means that your geographic range has no bearing on whether or not you go extinct at any moment in time. If you're below it, which is sort of a weird thing to think about, having a wide geographic range actually makes you more likely to go extinct. So something that has a broad geographic range today would be like, you know, a field mouse, rats. Okay, rats are everywhere thanks to us, but they have a very broad geographic range. Think about how hard it would be to make the Norwegian rat go extinct. Okay, it would be really, really challenging. Whereas an organism that has a very small geographic range that only lives in, say, you know, a certain tree in a certain part of the Amazon rainforest, it would be much easier for you to decimate that species. Okay, so that's sort of the logic there. So in background times, a wide geographic range reduces the chances that you'll go extinct. So here's some background time here. In between the little arrows are the mass extinctions event. Here's a good example where you're above the line. Having a broad geographic range means that you're much less likely to go extinct. But at most mass extinctions events, it doesn't matter at all. Having a broad geographic range does not help you. So that's another thing that sets these mass extinctions apart from background extinction. They defy this rule. Doesn't matter if you're super widespread, you're still at risk for extinction. There's a couple exceptions, and I'll talk briefly about one of them. The Ordovician extinction here doesn't quite conform to that rule, but the rest of them do. Okay, one more way to look at these mass extinctions, and that is in proportion of change of diversity. So here it's the same time scale. If we're below the line, it means that our diversity is decreasing, and if we're above the line, it means our diversity is increasing. So again, I've highlighted the 5 major mass extinctions. But I'm going to pull them apart a little bit. And what we're going to do, I'm going to use the Triassic as an example, is if we look at, we said that earlier, diversity is the net result of new species being created, origination, and then species becoming extinction. So it's the net sum of origination and extinction. So that means that you can get an overall decrease in diversity by having a pretty normal extinction rate and just not making any new species. Okay? And that's exactly what's happening at the Triassic. So what I've highlighted here is that the purple line is the extinction rate, which is slightly elevated at this boundary. But the orange line is the origination rate. It turns out that at this extinction event, origination was really, really, really low. How do you prevent organisms from originating, from speciating? It's a really great question that we don't really know the answer to. And so we call these mass depletions. All right, so we're depleting. So the extinction rate isn't particularly high. It might not actually be that much higher than background. And yet we're still getting a decrease in total number of species or genera because we're not making new ones to replace the ones that are going extinct. The big 3 mass extinctions, which are the End-Ordovician, the Permo-Triassic, and the Cretaceous-Paleogene, which you'll hear again and again. These ones are all are called the big 3 because they all have highly elevated extinction rates. Okay, so their origination rates are negligible or maybe even a little positive, but their extinction rates, the purple arrows, are big. All right, so that's quite different than what we saw at the Triassic. Okay, so now we have had a little overview of how we actually look at these extinctions and what makes them stand out against background times. So now I'm going to give you a whirlwind tour through the causes of the big 3 mass extinctions, and then I'm going to talk about one of these mass depletion events. Okay, so when we talk about mass extinctions, we talk about 2 things. One is the trigger. So the trigger is an event that leads to lethal conditions. And a kill mechanism is the thing that actually kills the individual. Right? Because in order to make something extinct, you have to go around and kill individual organisms. All right? Sometimes we think about it in a very abstract sense, but like these things have to die in order for their species to die. Okay, so first we're going to talk about the End-Ordovician, which is the first major mass extinction that we see in the Phanerozoic. So until recently, there was a lot of controversy over this mass extinction, but recently, some geochemical work, so paleontologists, we very often collaborate with geochemists who can help us reconstruct environments. So this is a paper that came out of a collaboration between a paleontologist and a geochemist. And what they did was they used a new type of isotope system to figure out the temperature of the ocean across this extinction event and figure out how big the ice sheets were. That seems sort of impossible, and I don't have time to go into the details, but you can do it if you're lucky. So what they did is they went around and they actually measured the isotopes in the shells of various types of fossils. So they used the fossils, but they actually used them for their isotopes, not for the actual fossil. And they did this in Anticosti, which is an island in the St. Lawrence Seaway, in Cincinnati, and under Mississippi Valley, other places. So this is the time bin where we see the extinction. And you can see that temperature plummets and then pops back up over a few million years. So there was a relatively rapid drop in global temperature across this boundary that was also associated with a growth of ice sheets. Okay? So you guys are familiar with ice sheets now, right? Okay. It's feeling a little bit end-Ordovician-y outside right now. Okay, so how does that actually make things go extinct? So this is a little cartoon of what North America looked like in the Ordovician. The beige area would be above sea level. The palish area would be a shallow sea, and then the darker blue would be deep sea. Now, marine invertebrates love shallow seas, okay? Sunlight drives photosynthesis, all the critters are happy, it's very, very diverse. So all of these little dots represent different types of sediments that are preserved from this time period. So carbonate is blue. So think of like the Bahamas, okay? So like happy reefs in the Bahamas, it's warm, it's beautiful. And then this event comes along and it becomes really, really cold. Okay, so imagine like reverse global warming is happening. What's gonna happen to the ice sheets? When it gets cold, what happens to ice sheets? They get bigger. Okay, so if we get big ice sheets, what happens to sea level? Down, right? Right now we're worried about sea level going up, okay? But imagine if you built up the Antarctic ice sheet, sea level would actually decrease. And that's exactly what happened here. So all of a sudden, from this little time bin here to this little time bin here, we lose a huge proportion of these nice, happy, shallow, warm seas, and a bunch of things go extinct. So the kill mechanisms are things like the species area effect, which just means that the less area you have, the fewer species that you can sustain, loss of habitats. Reefs really do poorly. So here's our happy shallow sea and our nice little barrier reef. You drop the sea level, things aren't good. Turns out that there's actually low oxygen during this ocean in the Ordovician as well. Another neat little feature of this is that organisms that are restricted to tropical settings had a much higher extinction rate than those that were adapted to higher latitudes. So this is a little bit of a tricky figure, but what I want you to focus on is this point here, which is the percent extinction basically in the time period that the extinction took place for organisms that lived between like 10 and 20 degrees latitude. Okay? So they had a much higher extinction rate than organisms that were already basically living in cold places. That kind of makes sense, okay? So when you think of like your friends who moved to Florida, you can just think of this, okay? Okay, so moving on to the largest mass extinction in Earth history, the Permo-Triassic. This is one of my favorite figures. I think it's from the Smithsonian. This is Doug Irwin, who works at the Smithsonian Institution. He's a paleontologist. And so Doug is kneeling in the Triassic with some paltry few fossils. And then this is the diversity of invertebrates from the Permian. Okay. So it's estimated that 96% of marine invertebrate species went extinct. It hit land vertebrates very hard. So at this time, we don't have dinosaurs yet, but we have lots of large amphibians and large reptiles and sort of proto-mammals living on the continents. It's the only time that we ever really see insects get hit by mass extinction. Insects are extremely resilient. And marine animal diversity takes a big hit. So these figures are a little tricky to read. They're called spindle diagrams. So each one of these represents one clade group of organisms, and the width of it represents their diversity. The red lines in each of them is the Permo-Triassic mass extinction. So you can see that where I've put little arrows, so the trilobites, our cute little friends here, go totally extinct at the Permo-Triassic. Other groups of organisms like crinoids, which are these often called sea lilies, and brachiopods, these guys over here, take big hits. So they're still around today, but their diversity has been reduced hugely. So what is the trigger for this mass extinction? The consensus now is that the trigger was actually volcanism, volcanoes. But not volcanoes as we think of them, like exploding Mount St. Helens style volcanoes, or even Hawaiian style. What we think caused this is something called the Siberian Traps. So traps is a word for a giant volcanic event. So here's Siberia. And the Siberian Traps, so all this red area here, there's evidence of actually lava at the surface. Okay, so this is like half of Siberia was covered in lava, all right, over many millions of years. So the original area is estimated to be about 7 million square kilometers, 1 to 4 million cubic kilometers of lava. And it's estimated that so volcanoes release CO2. They do it all the time. That's part of the carbon cycle on our planet. The CO2 release in this event is estimated to be over 4,000 gigatons. So far in the last 150 years, we've released probably more like 180 gigatons by now. So imagine, like we're really worried about the global warming that has occurred from that. Imagine it, an order, 2 orders of magnitude more. So that's a lot of CO2. Of course, it took place over a much longer time, over hundreds of thousands of years, but it still had a huge effect on the Earth system. So global warming would have been a huge issue. CO2 poisoning could have actually been an issue for some organisms. And then there's this really interesting story that I don't have time to go into, but there's a big group of bacteria in the deep ocean that produce hydrogen sulfide as a byproduct of their metabolism. And there's some people who think that some of that hydrogen sulfide may have actually been released, certainly into the surface ocean and maybe even into the atmosphere. And hydrogen sulfide is extremely poisonous to all respiring organisms. But a slightly easier kill mechanism to think about is what we're worried about right now, which is that as you push CO2 into the ocean, it drops the pH of the ocean. Okay, so ocean acidification, something that we're concerned about right now with global warming, would have happened on a massive scale at the Permo-Triassic mass extinction. So here's the chemical equation here. CO2 plus water plus carbonate gives you this little bicarbonate thing. So here's a happy shell. This is a figure from NOAA talking about current ocean acidification. So happy shell, and then I don't know why they make the shell burst into flame, but the point is that as you increase or decrease the pH of seawater, it becomes much harder for organisms to create their shells out of calcium carbonate. Okay. So one of the neat things about the Permo-Triassic is that we actually see a really cool example of what we call selectivity. So who goes extinct and why? At many mass extinctions, we'll see a drop in diversity, but it's not equal across the board. Some organisms do better and some do worse. So at this mass extinction, what we see is that organisms that can't deal with low pH go extinct or reduce in diversity. And there's a lot of interesting physiological reasons why they can't. So it's all about like whether, how much control they have over their internal fluids, basically, and whether or not they can pump ions one way or the other. So things like brachiopods and crinoids, turns out they're bad at that. Whereas fortunately for us, vertebrates, fish, snails, arthropods, et cetera, are much better at it. So they persisted through. One of the other, echinoderms is sand dollar here. One of the other really interesting things about this extinction is that it took a really long time to recover. So here's a little cartoon of what the seafloor would have looked like before the extinction, and then this is what it looked like for millions of years afterwards. So depressed diversity for a relatively long time after the extinction. So there's a huge perturbation to the Earth system that took a while for it to rebound from. So if we look here, here's the PT. You can't really see it on the scale, but diversity doesn't get very high. Then we get this little end Triassic thing this mass depletion here. Yep.
While you're on this chart, I thought a definition of extinction was 75% of families, was it?
It's a percentage like relative to neighborhood.
I mean, in terms of the end of ordination or the KPG, that doesn't look.
Well, so but the total, but it's like from here, right? So here you're only at 280. So it's not relative to today, right? So back here, you're at 75% relative to like the time period before.
But isn't it the whole height of that chart underneath it? That it's
I see. So this is families. So what I told you earlier, I think was genera? Maybe I had it wrong. So it depends on, a family is a higher Linnaean category than a genera. So in order to make a family go extinct, you have to kill off many, 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 many genera. So when we look at it at the family level, it's not going to be 75% because it's a higher level. Does that make sense? Yes, this is families. Yep, that's the discrepancy. Yeah, yeah. Okay. Trucking along to the mass extinction that people are probably most familiar with is the KPG, or what used to be called the KT. And here's some of my favorite cartoon explanations for this mass extinction. Hopefully, this is Gary Larson. It's like my favorite Gary Larson cartoon. It turns out that many other things also went extinct at this event, not just the dinosaurs, but the dinosaurs are the most well-known organism that went extinct. So the accepted trigger for this mass extinction is a giant meteor hitting hitting the planet. So the annual probability of a meteor greater than 15 kilometers hitting the Earth is that small number. So about once every 50 million years or so, we can expect a large object to hit the Earth. So the last one happened about 65 million years from now. We're due, right? But it's a, it's a statistical argument, so we could go another million years, another 10 million years, yeah.
What does KPG stand for?
KPG, Cretaceous Paleogene. So all of the time periods get letters assigned to them. Cretaceous begins with a C, but the Cambrian got C because it came before. So they gave the Cretaceous a K. It used to be called the KT because the time period afterwards used to be called the Tertiary, but then they renamed it to the Paleogene. So now it's the KPG. Yeah. I got this email yesterday. I just had to share. Phoebe helped protect our planet from asteroids and comets. OK, so it sounds like a joke, but it's actually from a real nonprofit organization called the Planetary Society. And there's a guy at MIT whose name I'm totally blanking on, who's one of these volunteers. So there's volunteers who basically scan for near-Earth objects. They don't get paid for this. It's like a international network of volunteers who do this work. So if you are feeling generous, you can donate to the Planetary Society to do that. All right, so here's one of many artists' reconstructions of what this day must have looked like. Here's the large bolide. It would have broken up into smaller pieces, and the dinosaurs and things bursting into flame. The evidence for this bolide impact, it's a really fascinating story. So the evidence for this impact was first found in 1980 by a father-son team called the Alvarezes. And they first identified evidence for this extinction event in Gubbio, Italy. So I was lucky enough to go there last year with 10 of my students from Williams College. So here's Tina holding up a piece of rock from right before the extinction event. And here I'm holding an ammonite, which is like a relative of a chambered nautilus. And this is one of the groups that goes extinct. Ammonites were extremely diverse right before this, and they completely go extinct at this boundary, so not just the dinosaurs. So this is what the boundary actually looks like. So the Sharpie is pointing to this layer here, which is called the clay layer. If any of you are looking for an uplifting book to read, I highly recommend Elizabeth Colbert's The Sixth Mass Extinction book. She goes here, digs out a little piece of the clay layer and talks about it. It's a really wonderfully written book about mass extinction. So what's so special about this little layer? This is the Cretaceous down here, and this is the Paleogene here. This layer is full of all sorts of evidence for a large impact, but the real smoking gun was that it's full of the element iridium. Okay? And iridium is extremely rare at the Earth's surface because it's what's called a siderophile element, which means iron-loving. And so when the Earth was a big molten ball of stuff, goo early in its history, all of the heavy elements sank to the core. That's why we have an iron-nickel core. And they brought iridium with them. And so the surface of our planet is highly depleted in iridium. At the same time as our planet was forming, there's lots of other stuff in the solar system, meteors, asteroids, and dust, that didn't have that differentiation process take place because it wasn't big enough to be melted. And so that stuff has tons of iridium in it. So if we find, this has orders of magnitude more iridium in it than we would expect to find anywhere on the Earth's surface. So basically the only way that you can explain that concentration of iridium is that a large rock fell out of the sky. And they calculated the size of the bolide based on the concentration of iridium in this layer and our understanding of the concentration of iridium in meteorites. And so my students actually did this in a lab. It's a great calculation because it's like just very simple arithmetic, and they can roughly estimate the size of the impactor. So the impactor was probably about 10 kilometers wide. And so this was a paper that was written in 1980, and it was hugely controversial in the field. And it took a long time for it to become accepted. And one of the ways in which it was accepted was by finding the crater, which is here on the Yucatan Peninsula. And you can't see it at the surface, but you can actually see it in gravity anomalies. So this is, here's the coastline here. And here are these little concentric circles. Everything's been covered in sediment, so it's like it's buried. These little dots are the cenotes, which are those little limestone springs. And you can see that there's like a ring of them around the edge of the crater, which is pretty cool. So how does this actually kill organisms? Well, obviously, if you're anywhere near this, you're dead. But what if you're on the other side of the planet? Well, the force of the impact was so great that it would have blown up a huge amount of material. Much of that material would have come back down onto the Earth so fast that it would have ignited plants upon impact. So there would have been global wildfires. People argue a lot about other kill mechanisms. The sunlight probably would have been blocked for a number of years because of all of the ash and dust. There's also some evidence that where it hit was actually a carbonate platform, so full of calcium carbonate, and when you vaporize that, it would have actually ended up releasing a lot of CO2 into the atmosphere. So CO2 comes in as a player. So there's a lot of different things going on. Here's one artist's reconstruction of sort of the fireball effect, where here's where the impact hits, but on the opposite side of the planet, there would have been widespread wildfires. So this is pretty well accepted, except that in science, no book is ever closed. And so there's a small group of scientists who have maintained for many years that this isn't the whole story. And they had a paper published recently, and there's an article in the New York Times about it. So the idea there is that there's also this large one of these trap things that's about the same age as this mass extinction in India, called the Deccan traps. So here's the extent of those traps. And for a long time, no one really had a good date on the traps, because if they erupt afterwards, it doesn't matter, right? They have to be basically the same time. So there's some new ages. So here's the Cretaceous-Paleogene boundary, and these are all ages that have come out of new work done partially at MIT and partially at Princeton. So now it's looking like maybe these volcanic events, or sort of here's some over here that are before, did overlap the extinction. So now people are talking about sort of like a 1-2 punch, that maybe the volcanism sort of weakened ecosystems, and then the impact, it was just like a really bad luck thing. Just a lot of controversy over this, in part because there are impact structures that we know of that are quite large that are not associated with extinction events. There are also large volcanic events that aren't associated with extinction events. So it's not as easy as saying, oh, I found a crater, therefore there must have been an extinction. It doesn't always work that way. And here's the image that was in the New York Times article, which I really love the graphics. So here's the volcano and the dinosaur. Why dinosaurs went extinct and not mammals? Still a very open question. Birds, which are descended from dinosaurs, made it across. Why? Why did the birds survive and not dinosaurs? Not all dinosaurs were big. There were small dinosaurs, that were chicken-sized. So why didn't they make it through if it was just big things that died? So there's still a lot of open questions about the selectivity of this extinction event that have not been answered to anyone's satisfaction. Okay. All right, so now I'm gonna, we've talked about sort of the big three. Now I'm gonna talk a little bit about a new project that I'm starting on one of these mass depletions. So I told you about the end Triassic mass depletion already. But there's another one in the late Devonian. So this is right after that, the really cold extinction and before the volcano extinction. So this is another example of a failure to originate. So here's our extinction rate is just a little bit elevated, but our origination rate is quite low. And this happens over a more protracted period of time than some of the other mass extinctions, so it's a little spread out. And in some ways, this is my favorite extinction, not just because I happens to be the one I work on, but it's sort of like the underdog. Like no one really understands it. It hasn't got as much attention, so I sort of like that. And it's also really fascinating to me. It's easy to understand how you make things go extinct. It's much less easy to understand how you decrease the rate at which new species are formed. So I think it's really interesting to think about how you create a world in which that happens. So fortunately for me and my collaborators, this is what North America looked like in the Devonian. So lots of shallow seas. This star is upstate New York. So we were in the Appalachian Mountains were really big at this time, like Himalaya, Andes size, basically. And they were shedding all this sediment into this ocean basin that's now upstate New York. And so for those of you who have driven across upstate New York and stopped at a road cut, the rocks are just full of fossils. And many of those fossils are Devonian in age. And so here's the wall of one of the many creeks that cut through upstate New York. And this is basically just a huge stack of sedimentary rocks that are Devonian in age. They're dark, organic, rich shales that were deposited in a deep inland sea on the west coast of this or sorry, the west side of the Appalachian Mountains. And many of these creeks expose this extinction event. And so in collaboration with a colleague of mine at Syracuse and another colleague at SUNY Oswego, I've started investigating this extinction event. The extinction event is expressed in these two really thick, stinky beds of black shale. They actually smell like gasoline if you put your nose up to them, because they're so organic rich. Okay, so gasoline, oil is basically just dead organic matter that's been sitting in a rock for millions and millions of years. So they're called Kellwasser events. They were first described in Germany. And there's two of them that encompass this extinction event. And this is what they look like. And they're very finely laminated, which means that they weren't bioturbated. Bioturbated just means something burrowed through them and mixed up the sediments. So if you see very fine layers of sediment, you know that there were no worms or other organisms burrowing in that sediment. And it's actually a really good proxy for the amount of oxygen there is. Because when the sediments are really, really low oxygen, the animals don't want to go in them because they're deadly. They're full of hydrogen sulfide probably, and they're not going to be full of food. And there may be no animals to burrow in them at all, because if they're that low oxygen, there's probably not a lot living in that area of the ocean anyway. So all of the extinction events that I've talked to you so far, we've been focusing on things like animals and to some extent plants. So for this extinction, the extinction of animals across this event is relatively well documented. But what's not well documented is what's happening with other parts of the food web. And so what I work on single-celled fossils. Most of what I do involves things that you cannot see with the naked eye. We dissolve rocks in acid, and we take the residue and we look at them under microscopes, and we can see fossils like these. Each one of these is like a tenth of the width of one of your hairs. So we measure them in microns, in thousandths of a millimeter. They're very, very small. But these are original organic structures that have been preserved for hundreds of millions of years. What are they? Good question. Many of them are probably the resistant coatings of single-celled algae. Some of them may actually be resistant coatings of eggs of animals. But the great thing about these microfossils is that the algal component represents sort of the base of a food web. And they're much, much more abundant than animal fossils. So in a piece of rock that we pick up from a creek in upstate New York that's like the size of my hand, I can find thousands of these things, whereas there may be 0 animal fossils in that same specimen. They have a very continuous record. So the animal fossil record is relatively spotty, so if you're trying to do really high resolution work, it can be very hard, whereas these things are very continuous. They're also less prone to issues with preservation, so they're more likely to get preserved. Animal fossils there can be different issues where you might be missing part of the record. So this is a brand new project. I just started it last summer. We've only looked at a couple of sections. So here's a section that we measured out in upstate New York, and these dark areas represent these 2 Kellwasser events. And what I'm showing you here are just some photos that one of my students took of some of these microfossils, just to show you that there are fossils throughout this entire event. And that's kind of interesting, because the animal fossils go away. But we do have this continuous record of microfossils through this extinction event. So these things aren't going extinct, which is, in a way, actually nice, because we can look at how they're changing, how the species are changing, how their abundance is changing. And we can use that to try to get a better understanding of what the environments were doing during this time and how that might have been affecting other parts of the Earth system. So one of the observations that we've made so far, which I'm not showing you the data here, is that the microfossils actually increase in size through these events, which is sort of curious. So this summer we'll be spending more time in upstate New York documenting the microfossil record across this extinction event and tying that into evidence from the geochemical record to try to figure out, okay, how is this extinction working? There's low oxygen, evidence for low oxygen in this area, and then thinking a little bit more theoretically about how low oxygen conditions may contribute to a decrease in the rate at which species are being created. So what drives periods of low diversity in Earth history? All right, other than rocks falling from the sky and volcanoes erupting, how is depressed diversity created? And how do environmental conditions, including low oxygen, depress diversity? And then the other interesting thing about this extinction is that it involves a decrease in diversity that's associated with low oxygen. And so some of you may be familiar with these things called oxygen minimum zones or dead zones. So these are places in today's ocean where oxygen has become so low that there's big fish die-offs, okay? And that oxygen is low for a variety of reasons. Oftentimes pollution and runoff can contribute to this. So all of the red areas here. So one of the reasons I'm interested in late Devonian is because it serves as a nice little analog for these oxygen minimum zones and these dead zones that we're seeing today. Okay, so now that I've sort of segued us into the modern, are we experiencing a sixth mass extinction? Many species have gone extinct since we've sort of expanded across the globe. Here's just a few of the sort of poster children of the extinctions. And it turns out to be extremely hard to figure out if you're in a mass extinction when you're in the middle of it. So it's easy for us to go back and say, okay, over this 1 million year period, we lose all this diversity. But can we say that we're in a mass extinction by just looking at the last 100 or even 1,000 years? It's really hard to say. So this is a paper that came out a couple of years ago asking, has the Earth's sixth mass extinction already arrived? And what these folks did is they looked at some major groups of organisms, so bivalves, things like clams and scallops, gastropods are our snails, amphibians, reptiles, birds, mammals, sharks, decapods like lobsters. And they tried to look at the percentage of things that had gone extinct. So the white is the percentage of organisms in these groups that we know have actually gone extinct. The black are the percentage of groups that are, of genera that are threatened, okay, or endangered. And then this black line is our 75, okay? So this is in species here. So each one of these little cartoons represents one of our big 5 mass extinctions over here. So the PT is way out here. So if you just look at the white numbers, you can't really tell a lot. If you look at the black numbers, then you could say, okay, maybe we're on our way to the 75% mark. But one of the tricky things about this sort of assessment is that for many of these groups, like say the bivalves, we actually know very little about the diversity of bivalves globally. All right, we're very familiar with some of them. The only clades in this chart here that we are probably accurate is the mammals and maybe the birds, because those are the organisms that are most well documented. There's sort of hidden diversity in all of these groups that we don't actually understand particularly well. So it's really hard to say exactly where these numbers are. But there is no doubt that extinction rates are elevated and that they're probably elevated against background. Does this mean that we are heading for a 6th major mass extinction? I don't know. And I don't think anyone will know for sure for hundreds, if not thousands of years. However, we can look at what is causing extinction today. And we can look at that in context for these previous mass extinctions. So this is a nice chart put together by my friend Paul Harnick, who teaches at Franklin and Marshall College, another liberal arts person. And here's the Ordovician extinction up here, which we think was caused by cooling and habitat loss. The little black dots there's strong evidence for that as being the cause of the mass extinction. The late Devonian, there's evidence for cooling, evidence for anoxia. The end Permian gets lots of dots, habitat loss, acidification, anoxia, et cetera, et cetera. Cretaceous Paleogene, evidence for acidification. And here's the modern down here. So the modern down here, there's evidence for acidification, anoxia, warming, and habitat loss. And then our unique human ones, overexploitation and pollution. But I think the key thing to know here is that the extinction that we're in now does share some drivers and threats with past extinctions. And so by understanding those past extinctions, we can actually learn a lot more about what we might expect to happen as climate continues to change. So an easy example of that is, of course, the whole ocean acidification thing that I talked about earlier. So can we use natural lessons from the Permo-Triassic mass extinction to understand which groups of marine organisms today are going to be most affected by ocean acidification? And will that give us the tools potentially to take to policymakers to try to protect the most fragile ecosystems and organisms? Okay, so now that we've talked about mass extinctions in sort of a negative light, I'm going to talk about them in a slightly more positive light for the end of the talk to sort of lift your spirits before you go off into the night. So mass extinctions turn out to be hugely important for creating the world in which we live in today. Okay? Without every single one of those mass extinctions, the world that we inhabit would look totally different. We probably wouldn't even be here. So what do I mean by that? So this is a diagram showing ecological patterns in, again, this is in marine invertebrates living in the ocean. So here's 541 million years ago, here's today. So this is looking at the distribution of organisms that live in different habitats. Oops, where's my, there we go, okay. So pelagic means that you swim up in the water column, so fish and whales. Mobile epifauna means you live on the seafloor, epi, on, but you can move. All right, so here's a little crab. Stationary epifauna means that you live on the seafloor, but you can't move. It's a thing called a brachiopod, which is sort of like a clam, but more boring, it just sits there. And then infauna means that you burrow into the sediment. So those of you who have ever gone digging for clams, who here has dug for clams? Yay, it's really fun. Okay, so they burrow into the ground, into the sediment. Okay, so they're infaunal. So as you can see, the proportions of these have changed over time. And I've highlighted the mass extinction events here. And so the most dramatic one is the Permo-Triassic. In pretty much every metric, the Permo-Triassic is the most dramatic. But you can see that there's a huge change in the distribution of where organisms are living at this extinction event. There's also a pretty big change right here, too, although this is the one that seems to be the most lasting. So these extinction events often restructure ecosystems at this sort of a level. They also restructure them in terms of which groups of organisms are most dominant. So this figure up top, the black line is extinction rate, and the little extinctions are labeled. And down here, this kind of crazy colorful graph is the same time scale. Each bar represents a bin of time, and the different colors correspond to different major groups of fossil organisms. So our gastropods are green. This blue here are our brachiopods. This yellow over here are scleractinian corals, which are the corals that make up our reefs today. So corals have been around for a long time, but the corals that we think of as being like coral reef corals haven't been around that long. What else? Oh yes, the red are cephalopods. That includes squid. So those ammonites that I was telling you about, very, very diverse group related to modern squids. The reason why this cuts off here is because that's where a lot of them go extinct. Okay, so if we highlight the Permo-Triassic and the KPG, we can see big shifts in the dominance of organisms, especially at the KPG actually, which is kind of interesting. We saw big ecological shifts back here, but in some ways we're seeing big shifts in the dominance of organisms right here. And another sort of infamous example of this is mammals. All right, so mammals coexisted with dinosaurs for millions and millions of years. In fact, mammals and dinosaurs originated at almost the same time. But the radiation and the increase in diversity in mammals did not really take place until after dinosaurs went extinct, a few million years afterwards. So this idea of sort of ecological incumbency, and that dinosaurs were filling this niche. They were the big terrestrial herbivores, the big terrestrial carnivores, and mammals were doing fine, they were doing their thing, but it wasn't until those dinosaurs went extinct that the mammals themselves could become the large terrestrial herbivores and the large terrestrial carnivores, for example. So the other great thing about mass extinctions is that right after a mass extinction, you see a huge spike in origination. So this little dotted line here is origination rate. So you can think of this as sort of like rising from the ashes. You know, after every large extinction event, there's an opportunity for new organisms to evolve, to radiate, and to fill these new niches. And so every mass extinction is followed by this big renewal. And one of the clades that appears after the KPG in this burst of origination are primates. So this is a reconstruction of a 55 million year old primate. So about 10 million years after this KPG mass extinction. Okay, so if not for this extinction event, primates may never have evolved. Okay, so what are the lessons for the future? No matter what we do, life will recover. It just might take a really long time. And I take weird comfort in this. Some people think it's like creepy, but it's, you know, we have this very sort of human-centric view. It's like, oh, we're killing the planet. It's like, no, no, no. We're probably killing ourselves, but the planet will be fine. Just give it a few million years. The planet will be fine. Life will go on. Life has always gone on. It has survived huge cataclysmic events, and it has continued and done really fascinating things every single time. Another important lesson is that all parts of the Earth system are connected. So we have these volcanoes that are changing ocean chemistry and maybe creating hydrogen sulfide from a group of bacteria that are then leading to the extinction of some organisms that have different physiologies. It's all connected, which is, I think, the best part of my job is sort of teasing apart those connections. Very fun and very interesting. And also that a faithful reading of the past can perhaps help us to see the future and hopefully give us some of the tools to change it. All right, that's it. Thank you.
This is about your own work, and I'm curious how you tell species for the microfossils, because you don't have any genetic material, I would assume, and so it's
That's right.
all shapes.
Yep, so DNA does not last very long. DNA degrades extremely quickly, so none of the fossils that we work on have any DNA left. People who work on much, much, much younger stuff sometimes get lucky. So we use morphology, the shape of the organism, to determine things like species level. With the microfossils, they may look like little blobs, but they actually have a lot going on. There's actually a lot of morphology there if you look at them closely. So I use a lot of different types of microscopes, light microscopes, electron microscopes, to try to understand that. At the end of the day, you do as much as you can with what you have. And that's, that's my lot in life for having decided to study these things, I guess. I like them, but yeah.
These microfossils, how differentiated are they? Look, they have systems. Do you see an alimentary system, a respiratory? What do you see?
They're like amoeba. So it's like a fossilized amoeba. So it's a single cell. None of the insides of it are preserved. So all we're seeing is the external morphology. Sometimes they do make hard parts. So there's many, many single-celled organisms today that that biomineralize. They make microscopic shells out of minerals. So some of the most dominant plankton groups in the oceans today, the coccolithophores and the diatoms, make shells out of different types of minerals. And so those also can get preserved in the fossil record.
Like a single cell.
But they're single-celled. Yep. Yep. Mm-hmm.
This morning on public radio, there was a piece that went a little too fast for me, but it was talking about changes in ocean level, like off this coast, it's gone up 5 inches.
Mm-hmm. Mm-hmm.
Actually, and in the west coast, they talked about the place where there's upwellings, which can lead to, I do have a question in here. But,
no worries.
Upwellings that can lead to more
to blooms. Yep.
More material
yep.
coming up and more life. But then there could be more CO2, and there were other issues, other things that seemed to negate positive effects. I don't know if you could talk about what you think.
So on the west coast of the Pacific, well, west coast of the United States, so the east coast of the Pacific, there's upwelling, which basically means the ocean currents are bringing lots of nutrients from the deep ocean up to the surface, and so they drive lots of productivity. So plankton, and the fish eat the plankton, and then they're all happy. So I didn't hear the piece, but it sounds like that's a positive thing, but
the pH was one of them.
But pH can also depress that. So decreasing pH will have a negative effect on ecosystems, no matter where you are.
And it said it was getting, I remember as you talked, the water was colder, which could then hold more carbon dioxide in the water, as which.
Yes, so the saturation state of gases changes according to the temperature of the water. So, so the colder water will hold more gas that will be dissolved in it.
So is there some understanding of whether the negative effects?
Oh, like which one will win out?
Yeah.
Yeah, I don't know. I mean, over the long term, global warming will win out if it proceeds unchecked for hundreds or thousands of years. Organisms have some ability to adapt, but evolution doesn't happen on the same timescales as human-induced climate change, for the most part. So, yeah.
What are the contemporary equivalents of the microfossils?
So, like I said, there's a ton of organisms in the oceans today that are single-celled that make. So there's algae that live today that make little hard parts out of. Exactly. Yep. Not exactly alike, but very similar. So I study the modern ones to better understand the fossil ones as well. So if you were to scoop up some ocean water and look at it under a microscope at the high enough magnification, you'd see all sorts of neat single-celled organisms that make structures that would potentially be preserved in the fossil record.
Thank you so much.
Thank you.
1940 China population, 520 million. Today it's 1.4 billion.